Um, dear audience, I would like to present my results, and I'm working at University of Gdańsk. Mainly, we were interested in bacteriophage TP84, and today I would like to say a few words about cloning and expression of thermostable TP84 bacteriophage endolysins. And construction of its fusion derivatives and prospective application. Thermophilic bacteriophage are rarely studied, and one of their life cycle have been known to a degree similar to Escherichia coli viruses. They are interesting object for studying determinants of thermophilicity and for practical aspect of biotechnology process using high temperatures. The project involved experimental and bioinformatics research concerning TP84 bacteriophage genome and coded proteins. Its 47 kilobase per double-stranded DNA genome relieved the presence of 81 coding sequence, as you can see here, for polypeptides of 4 kilodaltons or larger. Interestingly, all coding sequences are oriented in the same direction, and based on a homology search, a hypothetical biological function could be assigned to 31 coding sequence. Larger bacteriophage with double-stranded DNA encoded at least 2 lytic proteins, choline and endolysin enzyme with more lytic activity. At the final stage of the lytic development, choline phage causes permeabilization of the cell membrane, which allow endolysin to enter from the cytoplasm to periplasm and damage the peptidoglycan layer, as you can see on the left side of the picture. Proteins are produced in the late stage of infection. However, function are strictly controlled. Cholines are small protein that accumulate in the cytoplasmic membrane without harming the cells during the expression of late phage genes. They cause the hole in the membrane and initiate lysis by allowing passage endolysins. It exists the second lytic system, including endolysin with signal arrested release, SAR sequence present at the N-terminus end of some bacteriophage endolysins allow them to pass through the cell's membrane without the cholines, using only bacterial secretory system. Endolysins remains inactive until it folds properly after complete release from the cell's membrane. And the release from the cells of bacteriophage grow it's necessary from them to infect the next bacteria. The main barrier to overcome is the layer peptidoglycan. And our interest was the endolysin TP84-28 protein. It contains a domain responsible for enzymatic activity. Located at the N-terminus and a domain that allow for specific binding to selected sugar of the bacteria cell wall at the C-terminus. This slide shows the growth curve of Geobacillus thermophilus strain 10. It's a host of the bacteriophage. And here, as you can see, the orange color uninfected bacteria and gray color infected bacteria with TP84 bacteriophage. The optical density of the infected culture drops sharply from 80 minutes after the addition of the phage. We were able to obtain a pure preparation of native endolysin protein by purifying cell lysate using 3 purification steps. As you can see here on the left figure, the right figure. And the protein was the protein identity was confirmed by spectral analysis. To obtain a lot of active protein, the endolysin gene was amplified by PCR reaction using design primers and TP84 culture of phage as a DNA template. PCR product was digested with the enzyme BSA and SAL. Digested PCR product was mixed with a prepared factor and ligated into it by using T4 DNA ligase. The construct DNA was transformed into E. coli competent cells, and the recombinant clones were screened by colony PCR reaction, restricted analysis, and DNA sequencing. Positive clones was transformed into 3 different competence bacteria cells: E. coli BL21, E. coli Arctic Express, and E. coli BL21 Pelis S. The best strain of endolysin gene expression turned out to be E. coli BL21 DE3. The expected molecular weight of the recombinant protein is 44 kilodalton, as you can see. And once we grew in LB medium with ampicillin to optical density 0.8, and then induced by EBTG under the T7 promoter. As you can see on the left figure, the sample were collected after 1, 2, 3, 4, and 24 hours after induction and analyzed on SDS page gel. We observed the band on 44 kilodaltons after induction. After 4 hours of cultivation, cells were centrifugated and disturbed using ultrasonic homogenizer in buffer supplemented with lysozyme. And recombinant protein was purified from soluble cytoplasm fraction by precipitation. And after this, the mixture was heated to 55 Celsius degrees per 20 minutes to get rid of the mesophilic E. coli proteins. The supernatant was loaded on Ninta affinity resin, and after that purified on superdex resin. Protein identifier was confirmed by Western blot immunodetection with antibodies, as you can see of the figure of the right. Enzyme activity was measured by 2 options. First of one was standardized 2-BTD reduction assay, TRR, with host strain as substrate was used. Bacteria in the mid-exponential phase were harvested by centrifugation and were suspended in a phosphate buffer. Upon addition of enzyme to bacteria cells, the optical density was measured spectrometrically over time at 600 nanometers. And the second one on enzymatic activity of the proteins was determined in the demographic assay. Purified recombinant was fractionated by electrophoresis using SDS-page gel, including the Geobacillus teratromophilus strain 10. After running electrophoresis, the semogram was washed in excess of water and incubated at room temperature per 3 hours to renaturate the protein. And transparent bands arise where enzymatically active protein is located. Another option of the test is the fusion analysis, where we spotted the recombinant protein on the agar plate, supplemented with host cells Geobacillus strain 10. Data not shown here, but on another slides. I would like to say a few words about physicochemical properties of recombinant endolysin. Thermostability tests were also based on the endolysin activity and stability. We examined the measurement of relative activity under various temperature conditions. Activity assays showed non-significant reduction of the hemolytic activity after 30 minutes incubation of the enzyme in the range from 30 to 70 Celsius degrees. A significant decrease in the relative activity was observed at a temperature of 74 Celsius degrees. However, heating the enzyme in the temperature in the range from 75 to 100 Celsius degrees did not cause the activity to drop to zero, but only to reduce the activity to 33%. Therefore, we decided to measure the activity after treating the recombinant endolysin under autoclaving conditions, and this confirmed the loss activity to zero. The CD spectra of recombinant endolysin confirmed previous experiment that result indicates that secondary structure of this range from 30 to 75 is still has no changes, but when temperature was increasing from 75 to 100 Celsius degrees, the secondary changes, and here is noticeable on this graph. Differential scanning calorimetry analysis was used to determine the exact melting point and unfolding temperature of the protein. The melting point was found at 77.6 Celsius degrees. As you can see on the graph on the right side, clearly that the peak it's the denaturation of melting point of the protein, and furthermore, the characteristic appearance of the graph at 90 Celsius degrees indicates aggregation. Here, aggregation of protein molecules. In order to determine whether the construction of the fusion proteins would affect the activity of enzyme, we introduced a domain with a molecular weight of 17 kilodaltons at the N-terminus. The endolysin gene was cloned into E. coli system into a previously constructed vector with the fusion domain. And we conduct the PCR reaction of endolysin gene with starters including restriction sites for the sub-enzyme, then digested product with restriction enzyme and ligation with prepared vector. The expected molecular weight of the protein was 61 kilodaltons due to the adding a binding domain with molecular weight of 17 kilodaltons. Clones were grew in LB medium with ampicillin to optical density 0.8, and samples were collected after induction and analyzed on SDS page, as you can see on the picture on the left side. After 4 hours of cultivation, cells were disturbed using ultrasonic homogenizer, and the supernatant was purified on NINTA affinity chromatography. The fusion proteins was eluted in the range 300 to 400 millimole imidazole solution. We were able to purify the fusion proteins to homogeneity while maintaining the enzyme activity. And analogously to the recombinant endolysin, the fusion test was performed on Bacillus stereothermophilus for the fusion protein purification fraction. In the spot plates, there are transparent spots which indicate high activity of the enzyme. To control experiment buffers here and native endolysin, recombinant endolysin, and lysozyme were spots on the bacteria plate. To determine the host specificity of fusion endolysins, the lytic activity was tested on very gram-negative bacteria species and bacillus strains. Moralytic activity was measured by tuberosity assay and estimated via spot assay. Mesophilic and thermophilic bacteria as well, gram-positive and gram-negative bacteria were used to test lytic activity, and endolysin with a binding domain was investigated. Table show that activity was directed towards both gram-negative and gram-positive bacteria. Endolysin was mainly active against thermophilic gram-positive bacteria, including the host cell, and Bacillus stereothermophilus and Bacillus IC1. And then mesophilic gram-positive bacteria, such as Bacillus subtilis. We observed the enormous potential of the enzyme as an antimicrobial agent. Due to the modification of the protein, we are able to connect the enzyme with almost any form of medium interacts with the fusion protein. Due to patent reason, we cannot reveal the exact details of the binding domain. We studied the interaction of the fusion proteins with soluble medium using Blitz interaction. This was confirmed by the TEM spectroscopy, microscopy techniques, which is shown on the left of the right side. The fusion enzyme was combined with insoluble medium form, and the mixture was incubated overnight. After this, the mixture was rinsed with saline buffer and water, and then sent to TEM analysis. The connection between the medium and protein is clearly visible. All types of protein tested used spot assay. We claim that lytic activity is significantly observed on recombinant and fusion endolysins. We recognize that thermostable endolysin activity can be successfully used to disinfect surface exposed on high temperature, and potentially they are of importance as a replacement of supplement or for biotics therapy, as well for utilizing bacteria cells for biotechnology purpose. Thank you for attention.